We'll get off to a slow rolling start as people continue to trickle in from the waiting room. So hi, everyone. My name is Celeste. I'm the Patient Engagement Manager for the National Ataxia Foundation, and I am so excited to welcome you today to this talk on research and treatment development for SCA1. So 2 weeks ago, we had our clinical talk on SCA1 focusing on symptom management, which is available on the National Ataxia Foundation website. One of our lovely staff members who are in the chat will drop a link to that. But before we jump into today's talk, we just have a few housekeeping notes to go over. So first things first, if you don't mind or using the chat feature to chat with other attendees, just like you're doing now, in terms of if you want to connect with other people, share your thoughts and ideas about what is going on in the talk. If you want to connect with other people, please feel free to do that in the chat. Also, if you need closed captioning, you can turn that on. At the bottom of your screen, there should be a button that has a CC on it. You can click that from your menu and turn the captions on. Again, you can use the chat feature to chat with other attendees about what's going on in the talk. But if you have any specific questions, you can feel free to drop them in the Q&A window, which are also at the bottom of your screen here. So if you have any questions about the talk or questions you would like to ask during the Q&A session, feel free to drop them in the Q&A button. And then at the very end of the session today, we have some time to go through them. So again, if you have questions, don't put them in the chat. Please put them in the Q&A box. And with that, it is my pleasure to introduce our speaker today, Dr. James Orengo, who Dr. Orengo is a physician scientist in the Department of Neurology and Neurosciences at Baylor College of Medicine in Houston, Texas. He is focused on better understanding the mechanisms leading to genetic forms of ataxia and neuromuscular disorders. In particular, his research lab studies how brain cells important in muscle movement and strength, called motor neurons, become diseased in Spinocerebellar Ataxia type 1. So Dr. Orengo, the floor is yours.
Thank you so much, Celeste. And thank you to the National Ataxia Foundation for hosting this and inviting me. And also thanks to everybody in the audience for joining today. So I thought we could start with just a general outline of the topics for today, focused on spinocerebellar ataxia type 1. And it's a real privilege to be speaking with you all, and I'm looking forward to your questions that we'll get to at the end of the presentation. So we'll start out first with reviewing the biology of SCA1 and move on to how do researchers study SCA1, current ideas for therapeutic approaches, and then from a more practical perspective, what are the current research and clinical trials that are going on in SCA1? And then the National Ataxia Foundation, Celeste, will have a wrap-up session at the end. So let's start with the biology of SCA1. So first of all, it's a familial disorder, meaning that it's inherited in the family. It's a genetic disorder, and it's inherited in an autosomal dominant pattern of inheritance, which means that an individual with SCA1 has about a 50-50 chance of passing it on to any one of their particular children. The incidence of affected folks with SCA1 is about 1 in 100,000 people. And it's a progressive ataxia disorder, neurodegenerative disorder that leads to the loss of motor coordination. It also affects speech and balance. And this is primarily due to a slow degenerative process of the cerebellum, a part of the brain that is really important in motor coordination and balance and speech, swallowing. And in particular, within the cerebellum, there are these types of brain cells called Purkinje neurons that are highly vulnerable to the mutant protein in SCA1, as well as other brainstem neurons. And as you'll hear, there are other brain cells that are affected as well, lesser studied, such as what our lab focuses on, which is the motor neurons. And as the disease progresses, individuals will typically pass away prematurely from complications of respiratory insufficiency and aspiration pneumonia. So a little bit about the history of SCA1. The gene when mutated causes the disease SCA1 was first identified by two investigators, sort of independently, but working together from across the nation. One was Dr. Huda Zoghbi at Baylor College of Medicine in Houston, Texas, and the other Dr. Harry Orr at the University of Minnesota. And through a process of working with two families in their respective parts of the United States, they were able to identify this mutation, which is a unique mutation, and it is what's called a trinucleotide repeat expansion. So the gene in particular that when mutated causes SCA1 is called ataxin-1. And within ataxin-1, there are these CAG repeats in the DNA sequence. And if you have less than 39 CAG repeats, you don't develop SCA1. Whereas if you have 39 or more, you develop the disease SCA1. And in particular, the thing which causes the pathology in SCA1 is that this DNA is then turned into a protein. And the protein, the ataxin-1 protein, those CAG repeats, they code for an amino acid called glutamine. And so it makes these polyglutamine repeats within the ataxin-1 protein. And these polyglutamine repeats, they impart what's called the toxic gain of function for that protein. So they make ataxin-1 protein more stable, thereby aberrantly increasing its levels within the cells, within especially the neurons, the brain cells. And this prolongs its interaction with other protein interactors in a way that it normally wouldn't do if you didn't have the expanded polyglutamine repeats. It also promotes the accumulation and formation of these aggregates, these proteinaceous aggregates within the nucleus, which may be harmful for the neurons, or could also be a mechanism by which the cell just tries to deal with these toxic proteins. Well, as I was mentioning to you, SCA1 is a trinucleotide repeat expansion disorder, and these disorders are unique in the sense that most mutations are just either a deletion or an addition of some genetic material, or maybe a substitution. But these are repeat expansions. So here we have SCA1. It belongs to this special group of polyglutamine expansion disorders that includes other SCAs like 2, 3, 6, 7, and 17, and also includes Kennedy's disease or spinal bulbar muscular atrophy and Huntington's disease. And there are other repeat expansion disorders that can cause neurological disease. So for example, you can have another trinucleotide expansion, CTG repeats, in the most common form of adult onset muscular dystrophy, which is called myotonic dystrophy type 1. And you can also have hexanucleotide repeats, like these GGGGCC repeats located in this gene, C9orf72, which is the most common form of familial ALS, otherwise known as Lou Gehrig's disease. So these repeat expansions can occur in numerous different neurological disorders besides hereditary ataxias. So that's a little bit about the basic biology of SCA1. So how do researchers and scientists study this disease? Well, the main way they do is with model systems. And the most common model system used generally to study neurologic disorder are mice models, but others use rats as well. In the case of SCA1, really the bulk of all the research has mostly been done in mouse models. There are also human cell lines that could be used as a model system. And then there are alternative models to mice, animal models, that we can briefly touch upon today. So as I was mentioning, mouse models is really where most of the scientific research has been done to understand the basic mechanisms of disease in SCA1. And there are three main approaches that have been used. So earlier on, there was what's called a transgenic approach. But essentially what that means is that scientists took the human version of Ataxia1 and they popped it into a mouse. So they made a mouse that can express human Ataxia1 with various repeat lengths. Shorter repeats, that are thought not to be disease causative, and longer repeats, around 70, 80 repeats or so. So this has a advantage because you're using the human protein, human version of the protein, but it also has a disadvantage. And that's because the mouse is not used to making a human version of a protein. So there could be pathology in the mouse that's just related to a human protein and has nothing to do per se with the mutation, which is that polyglutamine repeat expansion. Also, when you do this, you have a little bit less control about the expression or the level, the amount of this protein that's being made in the mouse, because it's a bit more artificial in the way that it's introduced into the genome of the mouse. So you could have really high expression. In fact, you do have high expression of this human protein, and that in and of itself could be toxic. So the next kind of version, and the most commonly used model these days, is a knock-in model. So here, what scientists did is they took the CAG repeats that cause the disease, and they just knocked those repeats in, or they popped those repeats in to the mouse gene, the mouse version of Ataxia1. So this gives you a big advantage because now the mouse is making its version of Ataxia1 at its normal levels, at its physiologic levels. So there's not overexpression, and it's making it in the right areas, like the right cell types in the brain and elsewhere. But you have the mutation, the CAG repeats, which make that polyglutamine protein, and you can study the effects. And then the sort of next level after that scientists have done is they've made conditional models based on this kind of approach of using the endogenous mouse gene with the polyglutamine repeats. So these conditional models allow you to just specifically turn on or turn off the mutant gene in whatever cell type that you're interested in, or whatever time throughout the mouse's life that you're interested in. For example, you could turn it on only in cerebellar brain cells if you wanted, and not elsewhere in the body of the mouse. Or with these conditional models, you could have the bad protein being made, and then when the mouse is an adult, turn it off if you wanted to, and see how does the brain recover after removal of that toxic protein. So this is just kind of a broad overview of how mice have been used to study Ataxia1. And there have been hundreds of publications that have come from this mouse work, and lots of basic molecular biology that's been learned about Ataxia1, and also mutation in Ataxia1, that polyglutamine repeat expansion. So here's just a little bit a flavor of some of the highlights of the research that has come over the last 20 years or so in SCA1. So, well, the first thing that was learned, as I mentioned to you before, that the disease is a toxic gain of function. Well, that's important to figure out, because it tells you so much more about what kind of therapeutic approaches you might want to use to combat the disease. So how did the scientists figure that out? They figured it out by doing, as I mentioned to you, popping in those repeats, those polyglutamine repeats, into the mouse's Ataxia1 gene. And when they did that, this is the mouse that they created. As the mouse aged, it had started having some muscle wasting, some curvature of its spine, but more importantly, ataxia. And this was tested in various behavioral assays on the mouse. These mice also have brainstem dysfunction, Purkinje cell loss, and a reduced lifespan. So they're a pretty good model of the human disease. And so another hypothesis that somebody could have early on was that this mutant protein might disable the ataxin-1 gene. But that was proved to be false, because if you go and remove the mouse's ataxin-1 gene altogether, you basically have a normal mouse that doesn't really have any SCA1 signs or symptoms. Now, this is a really busy slide. Don't get bogged down in the details, but this is just an excerpt from a paper that came out in 2011 in Science by Fryer et al. And why I want to highlight it is because it shows one of the most important protein interactors of ataxin-1, which is called Capicua. Capicua is a transcription factor, transcriptional repressor, and it binds very tightly to ataxin-1. And when ataxin-1 has those polyglutamine repeats in it, it binds even tighter to Capicua and stabilizes it even more, thereby allowing it to do its job more than it should. And that in turn causes cerebellar degeneration. And so here you can see in this figure A, this thing called open field assay, where a mouse is placed into a field and you just measure how much does the mouse walk around in that field over a given period of time. And you could see that the wild type mice or non-SCA1 mice are this black bar, but the SCA1 mice, which are the red bar, walk much less during that period of time in the field. However, if you reduce the levels of this interactor of ataxin-1, Capicua, if you reduce those levels by half, by 50%, then you see a rescue effect where the mice walk more and they start to approach what a wild type or control mouse would walk. Well, and these are various different behavior assays that were done that show the rescue of different signs and symptoms that are normally seen in SCA1 that are modeled in the mouse. Again, here from that paper, if you zoom in and look at the cells in the cerebellum, these cells, these big ones right here, are called the Purkinje cells or Purkinje neurons. They're very important in the disease process in the cerebellum. And you can see in a control mouse or wild type mouse, this is what it looks like. But in the SCA1 mouse, you can see a dramatic loss of these Purkinje neurons. And this is, the primary driver for ataxia, or that incoordination of movement. If you instead take these mice and cross them with that Capicua mouse, where you have half the levels of Capicua compared to normal, you can see a rescue. You can see that SCA1 mouse, when you reduce its Capicua by half, starts to approach more like a normal mouse would in terms of its Purkinje density. Oh, and also you can see in this what's called a survival curve, you can see there's about a 3.5 week extension in lifespan simply by reducing Capicua levels by half. More recently, and this year in February, in 2023, Coffin et al. Published a nice work in Neuron, where they took the particular area in the ataxin-1 gene that is critical for touching and binding to Capicua, and they mutated it so it could no longer interact with Capicua. And if you do that in and of itself, you can have a pretty robust expansion in the mouse's lifespan, whereas the median time of death for a SCA1 mouse is around 40 weeks of age. These mutant mice that can no longer bind to Capicua are able to survive 9 weeks longer. And also this year, by Handler et al. In Neuron as well, they made another mouse model where they mutated what's called the nuclear localization signal of ataxin-1. So this is an important signal that some proteins have that tell it to go into the nucleus within the cell. And if you just simply mutate that signal so that ataxin-1 doesn't drive itself into the nucleus, you can see that even if you have the polyglutamine repeat expansion, which is so toxic, if it can't get into the nucleus, there's a very dramatic extension in lifespan. So with these investigators and their mouse colony, their SCA1 mice, median survival is about 27 weeks old. But if they simply made that mutation that in the nuclear localization signal, they were able to go from 27 weeks to 49 weeks for survival. So that's one of the largest extensions in lifespan. That tells us a lot about the biology of ataxin-1 and SCA1 and how it must be playing an important role in the nucleus in terms of disease development. So I'm just going to touch briefly on this just to mention that besides mouse models, there are also human cell lines that can be used to model SCA1. And sort of the older approach that has been around for a long time, 50 plus years, is to take cancer cell lines and just, either transiently or stably knock in your gene of interest, ataxin-1, for example, with various mutations, various repeat lengths. And why scientists use these cancer cell lines is that they divide indefinitely in a petri dish. So you can just keep on doing the work forever without the cells dying because they keep dividing because they're cancer cells. But as you can imagine, using cancer cells to study a neurological disorder maybe doesn't make the most sense. And as technology and science have evolved over the years, there's a new approach that's being used and is now the predominant approach in studying neurodegenerative disorders using cell lines. And that's called the iPSC-derived cell lines. So here's just two papers. I just did a snapshot of two recent papers that have come out where they made SCA1 neuronal cell models. So here what happens is that stem cells are isolated either from blood or from skin biopsy samples. And those stem cells are then differentiated in the laboratory to become neurons in a dish. And so here you're actually studying from an individual patient, from their stem cells, you're studying differentiated neurons in a dish. So this is the closest thing we can get to studying the disease in a human being right now, at least in terms of on a cellular and molecular level. So finally, about how we study SCA1, I want to mention about other animal models. So there are lots of other animal models that can be used, including worms, monkeys, I mentioned to you earlier, rats. But there are these two that have been used quite a bit in SCA1. The first one are Drosophila, or fruit flies. And there are numerous studies over the years and publications studying SCA1 in fruit flies. And in particular, what the scientists do is they make these models where they express the human ataxin-1 gene with those expanded repeats, the CAG repeats. And they study eye development because that's a nice readout in fruit flies of neurological disease. So here's what a normal fruit fly's eye would look like. And then here's what a SCA1 fruit fly's eye looks like. And you can see kind of the loss of these things called ommatidia, those structured components of the eye. And then, for example, in this paper by Park et al. In Nature in 2013, they screened a bunch of different compounds to see if they could find things that would rescue the disease. And here's an example of a suppressor that rescued the normal eye formation, in spite of the fact that these flies were expressing the mutant ataxin-1 protein. Finally, I just want to touch on zebrafish. So there has only been one publication so far on using zebrafish, and our lab, my lab, is currently making a new model as well. This is unpublished data, and here you can see what a normal control zebrafish looks like, adult zebrafish. And here's a zebrafish where we have put in the human ataxin-1 gene with a repeat CAG repeats or polyglutamine repeats. And you can see it starts to mimic or look awfully close to how the mouse model looks. So now I'd like to switch into the current therapeutic approaches for SCA1. Well, as we mentioned, it's a toxic gain of function mutation, those polyglutamine repeats in ataxin-1. So the top priority for many of the scientists in the field is finding out ways of reducing the levels, the protein levels of that polyglutamine ataxin-1. But there's some nuances here that we don't have time to dive into in detail today. But I just want to highlight some of the nuances. So if you reduce the levels, do you reduce all of the ataxin-1 level in the individual, or do you focus only on the mutant? Remember that each of us has 2 copies of every gene. One we get from mom, one we get from dad. And in individuals with SCA1, only one of those copies has the polyglutamine repeat expansion. The other copy is normal. It doesn't have that expansion. So if you develop a therapy to reduce the levels of ataxin-1, should you focus only on that mutant version of the ataxin-1, or should you just try to globally reduce all ataxin-1? Emerging science says it might be better to focus just on the mutant ataxin-1 levels. Although, as you can imagine, technically that's far more difficult to achieve than just reducing all ataxin-1 level. And another therapeutic approach, besides just reducing the levels, is to modulate those interactors of ataxin-1 that are thought to be important in the disease process, such as I mentioned to you earlier, that very important binding partner of ataxin-1, Capicua. And you saw that paper where they reduced the Capicua by half, and it rescued some of the symptoms of ataxin-1. So there are other interactors besides Capicua, and so one approach is going after those downstream pathways and modulating them, tweaking them in such a way that you improve the symptoms or the disease course in SCA1. And last, although this approach is not helpful for the particular individual with SCA1, it can be critical in preventing passing down the gene to that individual's children. So there is available family planning in which in vitro fertilization can be used, and obstetricians can select embryos which don't have that polyglutamine expansion to be implanted in the mother, thereby preventing any of the children from that individual with SCA1 from being born having that repeat expansion. So that's another approach to think about. Although it's a complicated one and an expensive one, it does exist at least. So let's dive back into that, what we were mentioning about just reducing the levels of ataxin-1. Recently, there's been a lot of excitement about this type of therapeutic technology called antisense oligonucleotide therapy. So here, a small compound, small molecule is made that can bind to any gene of interest and prevent its expression. So it's a way of reducing it can be a way of reducing the levels of a gene of interest. For example, in SCA1, Ataxia1. Why is this therapy so exciting? Well, it's exciting because now there are already 2 FDA-approved examples for other diseases. So there's a disease called spinal muscular atrophy, which is basically a form of ALS for babies. These small children and infants that have SMA develop progressive paralysis, and usually by 2 years of age or around that time, they may already pass away because they're unable to breathe due to the paralysis. But a few years back, the FDA approved one of these antisense oligonucleotide therapies that improves the expression of the disease gene in that case, which is called SMN1. And now these kids are living about 4, 5, 6 years old, and whereas before they were dying in infancy or as toddlers. So this has been really exciting in the field of neurology. And more recently, about 2 weeks or so ago, the FDA approved another antisense oligonucleotide therapy called tofersen that's used to treat a genetic mutation in ALS, a SOD1 mutation. So there's been a lot of enthusiasm amongst the SCA1 scientists about, could we use antisense oligonucleotide therapy to reduce Ataxia1 levels in SCA1 patients, and could that be an effective cure or therapy? Well, so far it's been studied in mice, and here you see this paper by Friedrich et al. In 2018, where they injected an antisense oligonucleotide therapy in SCA1 mice. And I'll just highlight, I know, again, it's another busy slide, I apologize, but this is straight out of the paper, the scientific literature. I'll just highlight the survival plot. So you can see that the red bar are the SCA1 mice that were treated with the antisense oligonucleotide therapy. And the blue line are the SCA1 mice that are not treated. The gold are SCA1 mice that were treated with a placebo, with a drug, but not the drug, not the antisense oligonucleotide therapy, just a vehicle, empty vehicle. So you can see there's been, in this case, it's days, the readout of survival, but there is a pretty significant increase in survival in these mice, and they have other rescue of other symptoms and signs of SCA1. So at least in mice, this therapy looks very promising. And others are working towards figuring out how to bring this into humans. Currently, there are no trials available, but this is an area of interest for sure. So that brings us to our last topic that I want to touch on, which is the current state of research and clinical trials. So here we need to have just a little bit of a background to understand that clinical trials come in 2 flavors. They can be therapeutic trials or observational trials. Therapeutic trials means you have some intervention, maybe a drug, maybe some device, or some change in clinical practice. And you want to see, does that intervention do something positive for patients with SCA1, either reducing their symptoms or curing them. That was what we all want. Versus an observational trial, there is no intervention in an observational trial, but there's just a detailed note-taking and record-keeping of how things progress. And also, these are important because they can identify what are known as biomarkers. They can identify things that can be easily found in the blood that might tell you how is a SCA1 patient progressing. And those biomarkers can be critical later when you have a therapeutic trial to see if you're having a positive effect or not. Besides, doing a neurological exam, sometimes it's nice to have a blood marker that can be followed. So if you're ever interested in finding about the latest clinical trials for SCA1 or for any disease, you should go to clinicaltrials.gov. And there you'll find a full listing of all the trials, those that have been completed, those that are actively recruiting, and those that are proposed and will be recruiting in the future. So this is a really great resource. So I will just highlight some of the things that are currently actively recruiting on clinicaltrials.gov. So the first one is a trial entitled a Safety and Pharmacokinetics Trial of V0659 in SCA1, SCA3, and Huntington's. So as I mentioned to you about the two flavors of clinical trials, this is a therapeutic trial. It's actively recruiting, and it's looking for individuals with SCA1, SCA3, or Huntington's disease. The goals of this trial is it's a first in-human clinical trial to assess the safety and tolerability of 4 doses of a new drug called V0659. An aim of this trial is to determine the concentration of the study drug in the cerebral spinal fluid and blood after 1 dose and after multiple doses. And the study drug is administered by lumbar puncture. So you need to have a spinal tap in order to get the drug into your body, as opposed to other drugs, which maybe you take as a pill that you swallow or an intravenous infusion. This one is what's called an intrathecal injection, which needs a lumbar puncture. What is V0695? Well, the preclinical data suggests that this drug may be a disease-modifying therapy in disorders through binding to the CAG repeats. Remember that mutation, those CAG repeats. And as those CAG repeats are expressed, this drug may help to prevent them from being expressed, interfering with their making that toxic protein with the polyglutamines. And that's why they're enrolling, not just SCA1, but SCA3 and Huntington's, which are all polyglutamine expansion disorders. If you remember from the beginning of our talk, there were 9 polyglutamine expansion disorders. And so the one benefit of this drug, if it works, is that it may be applicable to 9 different diseases instead of just 1 disease. So here, I'm not going to go through this in great detail, but just to say, it's always important with a clinical trial, if you're interested in it, to figure out what are the inclusion and exclusion criteria to see if you fit for it. And some of the highlights is that the individuals have to be between 25 and 60 years of age. They have to have a genetically proven SCA1, SCA3, or Huntington's with some kind of genetic testing. And they should have mild to moderate disease. And there are some exclusion criteria, like, for example, if you're pregnant or breastfeeding, that individual wouldn't be able to enroll in this trial. And if you have other mutations that can cause hereditary ataxia, you couldn't enroll in the trial either. Also, if you have any kind of thing that would make any other disease process where a lumbar puncture would cause more harm to you, then it wouldn't be able to enroll in this trial. So the site for this trial is in the Netherlands. Although clinicaltrials.gov is set up by the United States government, it includes all clinical trials, not just those that are being performed in the United States. So this is over in the Netherlands, and this is the principal investigator that's running this trial. So now let's switch gears to another clinical trial that's currently ongoing for SCA1. This is a natural history study of the genetic modifiers of spinocerebellar ataxias. So this one is not a therapeutic trial. Instead, it's an observational trial. And it's actively recruiting, and it's looking for those individuals with SCA1, 2, 3, and 6. And the goals are to figure out, how does the disease progress over time? What are the best ways of measuring the progression? And do some genes, other than the gene that's abnormal in the disease, have any effect on the way the disease behaves? It's a nationwide study. They're hoping to enroll 800 participants in the US. And the participants' enrollment will be for an undetermined period of time, meaning that they will be studying people long term. Study visits will be done every 6 or 12 months, depending on the participant site. And here's just a flavor, a sampling of the kinds of things they're going to be looking at. They're going to do blood collection to do DNA testing, study for genetic modifiers. They're going to do neurologic examinations, get some more history from the individual, do some functional rating scale measurements, such as walking 25 feet and hand dexterity, questionnaires about activities of daily living, and so on and so forth. So you can kind of see and collect demographic information, reviewing medical records. And here you can see there's lots of sites that are recruiting. So maybe there will be one close to where you live. You can see some of them that are recruiting, some of them that are active, but no longer recruiting, like this one in South Florida. So here's just a sampling. You can get more details on clinicaltrials.gov. Finally, I'll mention to you the last active trial related to SCA1, and that's this one, the Rare Disease Patient Registry and Natural History Study, Coordination of Rare Diseases at Stanford, or CORDIS. So this trial is not at all, it's an observational trial, just like the last one. It's actively recruiting, but it's not at all related solely to the SCAs or SCA1. Instead, it's related to all or many rare diseases. So that there are hundreds of possible diseases that they're interested in studying. And it's really more of a demographic collection or registry more than anything else. So you're not getting the detailed examination and detailed notes on the individual patient, but more just kind of a registry of individuals with rare diseases, including SCA1. And this one is being run, as I mentioned, by Stanford Health. And this is their contact information if you're interested in registering. So with that, I'd like to stop and say thank you so much for your attention. And I'm looking forward to any questions, and I'd like to turn it over to Celeste. Thank you.
Thank you so much, Dr. Orengo, for a fantastic talk, giving us a high-level overview of what's going on with SCA1 research right now. In terms of a couple things you can do next, if you want to stay up to date on the latest and greatest ataxia research news, you can become a member of the National Ataxia Foundation. You can get our newsletters to figure out and find information about what ongoing trials are happening. We have a participate in research page where you can find out about interventional studies, observational studies. You can join the CORDS registry, as well as different research studies that are going on. We also have support groups that are located all across the United States, Canada, and virtual groups across the world. So please feel free to access these resources. A couple of common questions that have been coming up that I just wanted to address before we jump into individual questions, which, as a reminder, if you go to the Q&A box at the bottom of your screen, you can type them in there. A few people have been asking if the recording of this video is going to be made available. Yes, it is. It will be available on the National Ataxia Foundation website in about a week. Also, we've been getting a couple of more technical questions from scientists in the Q&A. We're going to save those until the very end. We're going to prioritize patient questions first. So my apologies if we don't get to your questions. So starting off with a question about animal models. How many weeks in a mouse's life correlates to years in a human's life, or can that correlation be made?
Yeah, great question. Well, a mouse generally, the kind of mice that are used in the laboratory live for maybe a maximum of about 2 years. So if that helps to give some perspective, and they become sexually mature, able to reproduce as early as about 6 or somewhere between 6 and 8 weeks old. So we're talking about maybe a young upper adolescent teenage human being being around like a 2-month-old mouse maybe, and like a 6-month-old mouse being somebody who maybe in their 40s or so, perhaps. And then, something like a 1.5 to 2-year-old mouse being somebody closer to their 80s. That would be my best guess, but
No, that's fantastic. Jumping forward in the talk a little bit to a different topic, we have some questions about ASOs. So could you elaborate, does ASO therapy require lumbar punctures to administer? That is, do ASOs cross the blood-brain barrier? Could you elaborate a little bit on that?
yes. Currently, ASOs do not cross the blood-brain barrier well, effectively. So the only way to administer them is by lumbar puncture. And that's one of the challenges of rolling out those trials, is figuring out the dosage and the frequency, so that you get good penetration into the brain through those lumbar punctures.
Fantastic. And then we have a couple of questions relating to SCA1 and its connection with other kinds of disorders. So for example, if someone has a history of SCA1 in their family, or they have relatives that are diagnosed with SCA1, is there a likelihood that other people in their family may experience other disorders related to the cerebellum, like medulloblastoma? Is that common, or are those two things separate?
That's a good question. So yeah, definitely, if there's a genetically proven with a genetic test, the mutation that causes SCA1, then there may be other members of the family who may have SCA1. And as I was mentioning earlier, there's about a 50-50 chance of that individual passing it on, that genetic information, onto one of their children. In terms of are there co-associated other diseases that come along, to my knowledge, at least, there have been no links or data that show that there's some other disorder like cancer, for example, medulloblastoma, or any other disorder like heart disease or kidney disease in SCA1 individuals. So it's really focused in on that neurodegeneration in the brain, primarily in the cerebellum, but then at later stages in other areas of the brain as well.
That makes a lot of sense. We also have a question here about symptoms. Is there any connection between speaking symptoms and relating to physical balance, in terms of if you're doing well in one, are you doing well in the other, or SCA1 specifically?
Yeah. Well, in general, everything progresses over time. So as the balance gets worse, other areas like speaking and swallowing are getting worse too. But the remarkable thing is that each person is really a unique individual, and it's not stereotyped. It's not identical for everybody. So some folks may have more problems with speaking earlier in their disease. And maybe later on, they may have more problems with their arms and legs with the coordination. Whereas another person, the symptoms may first appear with their walking, and then the speaking problems may come on far later in the disease. So really, there are averages, and you can see how most people would progress. But for each individual, that you really are unique, and you may have a different progression than somebody else, even within your own family, that has the same mutation.
That's really good to know. We also have a question about, from the research that's out there, are there any protective factors that people can do to delay the onset of SCA1 in folks who have tested positive but are not yet showing symptoms? Is there any work on that?
Yes, there's lots of work on that, but nothing definitive that I can share. But yeah, lots of labs are interested in that, and resiliency, and either on a genetic level or through some kind of drug. What I can say more broadly, though, is that neurodegenerative disorders as a category, not just SCA1, but other SCAs and ALS, Parkinson's, Alzheimer's, all of those, there are three kind of core tenets that are important for slowing the progression of the disease. One is good nutrition. having adequate nutrition. Another one is regular exercise. And we're not talking about going to the gym and like bench pressing 400 pounds. We're just talking about some good low intensity cardiovascular exercise like walking or swimming or stationary bicycle, something like that. So exercise, nutrition, and rich social interactions. Having lots of interactions with family, friends, in a social environment and not being isolated. Those three tenets of managing a disease really does slow down neurodegeneration progression across the board. Another thing I would mention are antioxidants. So there are some foods that are rich in antioxidants like red wine, blueberries, dark chocolate. So incorporating those kinds of things into your diet, as we were mentioning, good nutrition and adequate calories, but enhancing some of those foods that are high in antioxidants may also be protective for those brain cells that are experiencing the neurodegeneration.
That's very good to know. I'm sure there's many people who are writing those down right now. Our next couple of questions are about clinical trials. So our first is, if you participate in an observational trial, does that mean you can't participate in a therapeutic trial or an interventional trial? Or can you drop out to join one of the others? Could you tell us a little bit more about that?
Yes, absolutely not. Yes. So if you are in an observational trial, you can still be in any kind of therapeutic trial. However, if you are in a therapeutic trial, you may not be eligible for a different therapeutic trial. Let's say there was another one besides the current one that's open. You probably would have to choose, between the two, and maybe being in one might disqualify you from being in another one in the future. But observational trials, no, you can be in as many of those as you like, and they won't play any bearing in any kind of future therapeutic trial you might want to get into.
It can be important to read through the inclusion-exclusion criteria when you're looking at therapeutic trials. Oftentimes they'll say if you've participated in others, you can't participate in this one. So something to keep in mind. The next question is about the VICO trial. So if a trial is happening outside of the US and it's going well, how long before it comes to the United States?
Yeah, well, that's a good question. I don't, to be honest, know the precise answer to that question. But we can give you an example. For example, in the world of ALS, there was a drug that came out called Radicava that was extensively used in Japan, and the major trial that got FDA approval came out from Japan. So there is a precedent for that. So if there was a tremendous success in this drug in the Netherlands, they could apply for FDA approval, and then if they get approval, then it would be available for all of us here to prescribe in the US.
Speaking of sort of future potential therapies, once a therapy has been approved by the FDA, would it be something that could help all stages of SCA1, or would it be more effective for folks who are asymptomatic, who are late stage, who are experiencing symptoms? Could you comment a bit on that?
Yeah, it may depend exactly on the specific therapy. So if the therapy is primarily working to slow the progression of the disease, then the earlier you can be on the therapy, the more bang for your buck you're going to get. So if you're asymptomatic or just very mildly symptomatic, and you start taking a medication that slows your disease progress, then you may get many, many months and years of extension because of that. But on the flip side, if you're already reaching sort of the end stages of your disease, then you may not get as much benefit from that medication. So that's something to weigh, with your physician about whether to start or not. How on the other side, though, if it's a therapeutic approach that doesn't slow the progression, but instead completely removes the offending problem, like the toxic polyglutamine ataxia-1 protein, then in theory, anytime you start it would be good. Even towards the end stage, you may not be able to get things back that you've already lost, but at least you will have halted the ongoing progression. So if that could work out and be an effective therapy, it would be really great, to reduce or remove altogether the mutant polyglutamine ataxia-1 protein.
Fantastic. And now we're going to jump back to the topic of IVF. So how effective is IVF for getting rid of the SCA1 gene in terms of, is that like 100% effective? Could you tell us a little bit more about IVF for SCA1?
Yeah. So the way that IVF or in vitro fertilization works is that you get some eggs from the future mom and some sperm from future dad, mix it together, fertilize those eggs, and you get embryos. And usually several embryos are generated. 10 or so or more are generated. And then those embryos are grown in a petri dish for some period of time. And very carefully, each individual embryo is sampled. Some of its genetic material is sampled, and a genetic test is done to see how many repeats of CAGs are in the ataxia-1 gene in that embryo. And any of the embryos that cross that threshold where they would develop SCA1, like they have 39 or more repeats, those embryos would not be implanted. They would be set aside. And the embryos which have less, which won't develop into an adult with SCA1, those would be the ones that are taken and implanted back in mom. So I don't know if I exactly answered the question, but hopefully that helped give some clarity.
Oh, that's definitely helpful. And then we're just having a few people ask for some clarification about how autosomal dominant ataxias are passed down. So if someone isn't experiencing symptoms yet, but doesn't know their gene status, does that mean they could still have the gene? Could they pass it on to their kids? Could you go over that again?
Yeah. So autosomal dominant is just a term, but basically what we're talking about is that in SCA1, all of us for every gene have 2 copies of every gene. So in SCA1, we have 2 copies of the ataxia-1 gene. And if one of the copies has these repeat expansions, the CAG repeats, then we can develop SCA1. So if that individual were to have kids, randomly, they're going to pass on one of their copies. Let's say it's a mom. She's going to pass on one of her copies of ataxia-1 to make that future baby. And dad is going to give one of his copies. Well, if mom has SCA1 and she has 2 copies, and one is a normal copy and one is a mutant copy, there's a 50-50 chance which copy she's going to pass to that egg. So if she passes the normal copy, then that kid will grow up and never have SCA1 and never have any different likelihood of developing SCA1 than anybody in the random world population. But if mom instead passes on that mutant copy of ataxia-1, then that kid, when they grow up, will develop SCA1. When they will develop it, nobody knows. It might be in their 30s, maybe in their 50s or 60s. As I was mentioning before, each person is a unique individual.
So it doesn't matter if the parent passing it on is having symptoms yet or not. It's always a 50-50 chance.
Correct. If the parent is established that they have SCA1. Yeah.
Well, that's all the time we have for today. But thank you again so much, Dr. Orengo, for walking us through SCA1 research and treatment options. My apologies to the folks we couldn't get to your questions, but thank you all so much for joining us today. And depending on where you are joining us from the world, good afternoon, good evening, good morning. Hope you have a wonderful rest of your day. Thank you so much. Bye, everyone.